Hey, what is going on? It is February 24th, 2022. Yeah, we're into hitting the home stretch for February, which I've been mentioning for a few days. And yeah, it's been a pretty crazy few days as far as the world is concerned. Right now, Russia is actively invading Ukraine. I feel like this is a harken back to the Cold War times, although I don't know exactly what happened during the Cold War in terms of conventional direct conflict between two powers, but I guess this has some equivalence to those proxy war type situations that happened with the USSR. But anyway, I'm not here to talk about proxy wars between human beings. That stuff is old hat, in my opinion. And what I would rather talk about today is, the start of oncogenetics and tumor suppressor genes. This is like a huge subject, obviously. Cancer is the leading cause of death amongst people, other than the leading cause of, I wouldn't say non man made death, but I would say natural consequences, right? Or natural, non like violent death, I guess. I don't know. Whatever the case is, the thing is, the goal is not really to go super in the direction of cancer, though we have to, right? To talk about the the extension of life, human life, because eventually I've been told that everybody suffers cancer and at some point in their life, no matter how old you are. And that makes sense because your cells no longer have effective cellular machinery, which prevents the processes that I'm about to talk about. But again, this is something that I am very, it's very nascent in my knowledge sphere, and so I'm going to have to revisit this a lot. But this is essentially close to where I wanted to get to, right? I really wanted to delve into the cell cycle and cell regulation because this is like one of the core components, as I mentioned yesterday, in the extension of human life, right? To essentially make sure the cell cycle faithfully continues on its activity course, and that applies, you know, for many things. Not only the cell cycle, but the cell cycle is a necessary, not sufficient, but a necessary area of study to really master in order to understand and figure out, I think, on how we can extend human life. And I'm sure that other people who are well versed in this with extensive oncogenetic backgrounds and life extension backgrounds will also mention that as well. The issue is that this knowledge is not very accessible to the average person. But anyway, I've ranted on for a long ass time, a long time, sorry, about, where I intend to go with this. So let's get on to it a little bit. So I'm just going to jump around a bit. And there are two, so the last time we were talking about the cell cycle, right? And the cell cycle is basically, this is the overall representation of the cell cycle. You have interphase and mitosis, right? And then interphase is broken up into the gap phases and the DNA synthesis phases, and the gap phase 1 can sometimes go into the resting phase, where there's no cell growth and the cell is just undergoing cellular activity. That is the longest phase for the more permanent cells and the stable cells, rather than the labile cells, which constantly have very short G phases and G0 phases, because they need to get through replicating fast, right? Epithelial cells, etc. So I also started talking about cell regulation, and this is not pictured here, but if we look at cellular checkpoints, that's important to always bring up. Cellular checkpoints. The reason why cellular checkpoints are important is because, and I talked about yesterday, these are where the so-called cyclin proteins work with cyclin-dependent kinases, which is another kind of protein, to phosphorylate other downstream proteins, which allow for the progression of the cell through its various phases, right? So the cell only knows, for example, to go from G1 to S after the checkpoint, G1 checkpoint has been reached and surmounted, right? And then, for example, going more specifically, the G1 checkpoint checks for DNA damage, right? If there's damaged DNA, we don't want to go synthesize that damaged DNA, right? If there's DNA damage, which is detected at the checkpoint during the growth phase, then we want to either correct that damage or we want to apoptotically get rid of the cell. Okay, so that's kind of what I touched on yesterday, and here's more of a in-depth diagram of what's going on there. So again, checkpoints, just reviewing yesterday a little bit of what I talked about, the checkpoints are related to the functions primarily of what happens during the phase. Like the checkpoint in mitosis is called a spindle assembly checkpoint, and that, remember the mitotic spindle, right? Like when the chromosomes line up during mitosis along the metaphase plate, right? That is the center of the cell, metaphase plate. See, this is the metaphase plate. There's a checkpoint which makes sure the chromosomes are lined up properly. Okay, so I don't yet know what the cell does to correct the situation where the cells aren't lined up on the metaphase plate exactly, but it is one of the things checked during the it is checked during this spindle assembly checkpoint, because I guess it's something that could be prone to quite a lot of error, right? So the attachment, there's probably tons of mechanisms to double check it, but this is the final checkpoint. And then the other checkpoint is after, this is the after mitosis. I'm going out of order a little bit. Then the G2 checkpoint checks for before mitosis in the gap2 phase whether the cell is of the right size, right? So basically, can the cell effectively divide into identical cells that have the same organelles and cytoplasm, etc.? So that's another important thing to check. So remember, all of these are related to the phase that these checkpoints are related to the phase that they occur in, or that they occur before, right? So anyway, let's get to the other piece of this. So what's happening here during these processes? Because so we have to talk about, and we'll get to this more, we have to talk about tumor suppressor genes. Okay, there's two categories of genes that we actually need to talk about: tumor suppressor genes and proto-oncogenes. Okay, and oncogenes. Tumor suppressor genes really are, it's a humongous category. It's where most people who are into oncogenetics spend a lot of their time, and I think most biology students need to understand a lot about it. It's a very complex, involved set of mechanisms that are involved in preventing, as the name is pretty simple to understand, tumors from forming. The actors in the whole tumor suppressor process, one of them, you'll hear these, I've listed out some of the common actors. One of the major actors are retinoblastomas, right? So it's funny because retinoblastoma gives you an indication of, like, makes you think of the eye, right? And the reason why these genes, retinoblastoma genes, are pretty much present in every cell. So why are they called retinoblastoma? Because they, I guess, were early on indicated as being prevalent in the cancer of the eye called retinoblastoma. And I have to look into the exact why a little more, but yeah, it can be looked up. And so retinoblastoma genes, retinoblastoma is a tumor suppressor gene, right? That essentially what happens is that it binds to a protein complex called E2F normally, right? It has the ability to bind to E2F. E2F is a protein which allows this, it allows the transcription of, it's a transcription factor. It promotes the creation of proteins from the DNA through translation to allow for the cell cycle to progress, right? So basically, what happens is, in order to prevent cell cycle progression, right, or to allow for cell cycle progression, either way, there's this RB gene binds to E2F and gets hyperphosphorylated, right? So hyperphosphorylation occurs via intermediates from cyclin-dependent kinases. Remember what kinases do. Protein kinase is phosphorylate proteins, right, and allow them to carry on extra activity. So basically, the hyperphosphorylation of this RB occurs so it can bind E2F, so that the cell cycle can be regulated, right? Because E2F being non-regulated allows creation of proteins which allow the cell cycle to progress. But the tumor suppressor gene, and that's why it's a suppressor, right? It holds onto E2F. But if the tumor suppressor gene cannot hold on to E2F because it's been affected by a mutation, and in when I talk about mutation, I'm talking about a mutation that usually comes from the genetic lineage of parents. Then what will happen is that it will ignore it will not bind to E2F. E2F will constantly go along its process of creating products which lead to the cell cycle continuing. For example, the S phase, continuing from the G phase to the S phase, will go unimpeded. So basically, normally what happens is there's many mechanisms downstream to prevent that from occurring. Like, for example, there's DNA damage checks that occur. Those genes we'll talk about later, but those essentially will also stop E2F activity from occurring as well. And in fact, what DNA damage checking is something that slows the cell cycle down. And ultimately, there's this magnitude of whether the DNA is so damaged the cell can wait and repair it before going into S phase, which will delay an S phase transition, or the cell will be signaled, apoptosis will be signaled. So that apoptotic signaling is what we're going to get to in the next tumor suppressor gene. So there's many tumor suppressor genes, but the big actors are the retinoblastoma gene, which is indicated in many cancers. Actually, I think about 50% of cancers, I believe, if I remember, have mutated retinoblastoma genes from both parents. Both alleles have been mutated, and those mutations can occur on the parental lineage, or in fact they can even occur spontaneously. Spontaneous retinoblastoma mutations, I'm not sure why they occur, but they can occur. The p53 genes, now here's the the king of tumor suppressors, right, as they are called. They're p53. So p53 essentially allows for cell cycle arrest, and it has many different functions, right? So it's the police officer. It will cause apoptosis of the cell, necrosis, necroptosis, ferroptosis, all sorts of programmed death will occur. Cell autophagy is also something that's indicated in life science studies a lot, that occurs as a result of p53 processes, senescence. It can activate all these things, right? And this is one of the final checks, right? And one can think, like, when an enzyme does all this, right? It's not, the enzyme has really no intelligence. It acts on factors. So the way that the body prevents enzyme overactivation is that there are regulators for enzymes. Even enzymes that regulate other processes have regulators for themselves, right? So p53 will go along doing things, right, to prevent the cell cycle from progressing should it determine that that there's DNA damage that will lead to cancer, right? That's what happens in a nutshell. And eventually, the E2F binding will not occur as a result of p53 involvement also, but sometimes p53 is over aggressive, so we have to actually wait to see if we can correct whatever p53 is marked, and we can then move further into the cell cycle. Okay, so that's p53, and then E2F is the last thing that I want to talk about. E2F is what I just talked about. It's a earlier when talking about retinoblastoma. It's a transcription factor. See, retinoblastoma protein binds to E2F, which E2F normally has consequences in that it allows for the creation of other proteins, the cyclins that are checkpoint dependent, right? Are involved also. They hyperphosphorylate RBF, which has a effect on E2F and allows E2F to either progress or not, whether retinoblastoma can hold on to E2F or not. So that's it in a nutshell, right? Like, I will clean this presentation up for the next time. There's a lot to talk about here. I went about 15 minutes today, and yeah, we'll get more into each of these guys at some point as well in the future. All right, that's it. If you like this, like, subscribe, and all that good stuff. I'll see you around.